Go for it.
Yeah, are we recording? The streaming? Okay, cool. So welcome everybody. Thank you for taking the time to join us for this seminar, this exceptionally busy week. I know most of us have been, quite zoomed out in general, but this week in particular has been a challenge to keep all the schedules moving. So really appreciate you taking the time to come here. Dr. Bomfim, who I have the pleasure to introduce. Barbara and I know each other for a long time. I'm a big fan of hers. She received her PhD in soils and biogeochemistry at UC Davis when I was a postdoc there at the time. And we go further back than that, but I'm sure she'll mention a couple older stories in her talk. Maybe not. I'm not going to be disappointed if not, Barbara. But she was a postdoctoral scholar with us at U of O for a couple of years. She led some very important projects in my lab. It was a pleasure to have her on board, and she's now a research associate at the Lawrence Berkeley National Laboratory, where she's developing what I consider one of the most innovative research programs I've seen anybody put together in recent years at the intersection of terrestrial ecology, soil biogeochemistry, with implications for modeling and some broader macroecology and evolution, if we will, at the intersection of climate change and terrestrial ecosystems dynamics. So I'm really thrilled to have Barbara here to speak for us today. And without any further ado, Barbara, the floor is yours. Thank you for accepting our invitation.
Thank you, Lucas. Okay, so I'll share my screen. And so let me know if you see the full version. Okay, good. All right. Hi, everybody. So as Lucas introduced me, I'm Barbara. I'm actually a postdoc at the Berkeley Lab, not a research associate yet. So I'm in the Climate and Ecosystem Sciences Division. So today we're going to talk about the hidden stories beneath our feet. So it gives you an idea that we're going to talk about soils. And we're going to look into the ecological and biogeochemical responses to ecosystem disturbance. So what we're going to see today, I'm going to go briefly explaining a little bit of my trajectory coming from Brazil now to Berkeley, and a bit about my research interests and some parallel activities I did as I received my education. So then we're going to jump to talk about fire. Many of you work with fire, so that should be cool for you. And then finally going to talk about the climate change experiment that I was very fortunate to participate during my postdoc time in Oregon. Okay, and feel free to write some questions in the chat. I know that sometimes we have a question when the person is speaking and then later we forget. So if you want to stop me, great. If not, you can throw something in the chat and then later we go over it. Okay. I don't know how you usually do this is as far as moderating, but whatever. Yeah. Okay. So I come from this forested country in South America named Brazil, right? In Brazil, I studied forestry, but my focus was more on plant community ecology, right? Not only savanna ecosystems, but also in forest ecosystems, and with a strong focus on conservation of the Brazilian savanna that we call the Cerrado. So this is a photo of me measuring a tree height. I did a lot of field work, and field research was really the world of research for me, right? As an undergraduate, it's not like I could choose between, let's say, computer modeling or field research, right? It's like field research was the world of research for me. So I embraced it, and it's something that still I carry, right? It's something very strong, and I still do, except now for the pandemic, right? So no field work now during the pandemic. And this photo here is just to show, Brasilia is a kind of modern city, very young. And this is the National Congress. People visit the city for architecture, right? More than just to see what's around in the Savannah. Then I did a joint master's between Brasilia, the University of Brasilia, and then the INPA, which is the National Institute for Amazonian Research in Manaus. So that's when I started getting more into soil thing. I looked into the effects of logging on soil in a sustainable forest management company. We're not going to talk about this, but it was the way that I saw that soils matter a lot, even if we think about forest or whatever terrestrial ecosystem. Then I came to California, to Davis for a PhD in soils and biogeochemistry, but all my research was done in Brazil, right? So I went back and forth to Brazil to do all the field work and sample collection, and I worked with fire and land use change. And the fire portion we're gonna see today. Then I went to Oregon and had the opportunity to look into climate change effects in Mediterranean prairies. It was the sort of the first time working with a non-woody type of ecosystem, right? So a lot of learning, and we're going to talk about this today. Then again, coming to Berkeley, not again to California, and this time to Berkeley, where I'm working the NG Tropics project. So it's a very big project. It's a sort of team science, which is the foundation here at Berkeley Lab. And I am doing the empirical work to help model development. And I'm looking at the role of soil fertility in shaping how forests respond and recover to cyclone, right? So different disturbances that I hadn't worked with before. So a lot of learning too. So I did a lot of parallel activities too during that time. So all these dots were the places in the world I traveled during my education. I competed in this strange sport named adventure racing that only 0.01% of the world's population know about. So there was a lot of explaining what's adventure racing. So I'm going to explain to you in 40 seconds. So this is a picture of us. It's a team sport. So it's usually 4 people competing across the landscape everywhere in the world. And we need to plan our routes. So there's a lot of strategy, a lot of planning because we use just map and compass. So no GPS allowed. And then we run, we bike, we kayak. It's a multidisciplinary type of sport. So that I did during my education, and it was very helpful, I would say, for grad students now that are struggling with like finishing grad school, right? I think it was an opportunity for me not to focus too much on school and be overwhelmed. So I always had the balance between them doing sports and my education. And many times when you're trying to figure out something in your research, it's maybe helpful for you to get out and do something, and it allows your brain to just relax. And then maybe you have the opportunity to understand exactly what you're doing, and then maybe even have an idea. So that was, I think very beneficial to me in some way. Of course, when I was on my bike, I was not publishing papers, but I was thinking about my research a lot. But then all that fun was substituted with my motherhood, right? This is a photo of me right off my PhD defense. I was very relieved to have one less thing to do, but I also had this big baby here to take care of, right? So it's very challenging to be a mother in academia, right? And, all those challenges I faced, being the only woman in a team with other 3 guys and then being a mother, make me think about gender equality, right? And I think about this more and more today. And I didn't used to think about that much in the past, right? So what I say is that one size does not fit all, right? Women have different needs, different barriers, right? So I think about that in all steps of my research nowadays. So based on this mix of a bit of everything that I did before, it's not surprising that sort of my research niche falls in between different areas, right? So what I am interested in falls at the intersection of ecosystem ecology, disturbance ecology, soil science, and biogeochemistry, right? So the blend of everything is, where I get inspiration to, think about research question, testable hypotheses, at least as much as possible. And the mode, how I have done research so far has been generating new data, right? So generating data, analyzing it, reporting it, right? Communicating, and then having the opportunity to refine questions, refine approaches, and keep going on. And then most recently, I've been working very closely with modelers. So I'm more aware of the challenges that modelers face. So I think about that now when I generate new data, right? So one of, say the guiding research question I have in my research is to understand how disturbances affect nutrient availability and nutrient controls on ecosystem processes. And for that, as we're going to see today, I've been using soil as a model system. And soil is interesting because it's an ecosystem in and of itself and also an ecosystem component. and an important one, right? I'm not going to have the full list of, why soils matter, but soils, they store carbon that fuel the activities of soil heterotrophs, like the ones that I investigated in my research, nitrogen fixers in the soil, and also holds nutrient elements, water, and provide physical support for plant growth and microbial activity. So, now you have seen an overview of, what I've done and my interest. So we're going to focus on the fire portion of the presentation. So fire in the Brazilian Amazon is different from fire in Western US, right? So that's why I'm going to explain a little bit about why fire in the Brazilian Amazon. So we have been seeing an increase in fire events recently in the Brazilian Amazon, right? It's not something I grew up with. I did not grow up hearing that fire events were dangerous or were somewhat negative in the Amazon, not at all. Right, so this region you see here, where this square is where you see this bunch of red dots, which are fire events detected on August 4th, 2020. And this area in yellow is where deforestation has happened, and the green is the remaining forests. So what's happening is that in that border where you saw the yellowish, we have a natural coexistence, right? So we have forests that are moist forests and flooded forests that coexist with savanna, right? The savanna type vegetations where my cursor is, I hope you can see it here. And then this darker green are the forests, right? So naturally fire has been present, right? But because the forest is moist, fire does not succeed in advancing into the forest, right? So sort of like dies off. However, with climate change, scientists have found that the dry season length in that region has been increasing, right? So longer dry seasons, right? Longer dry seasons mean longer fire season. And that has been a problem. That combined with deforestation, right? So increased fragmentation, you have more fragments and then you have edge effect, right? So these forests, they become drier. Then fire has become more successful in, ingressing into the forest than it used to be before, right? So all the combination of all that makes now fire a real threat to tropical forests in the Amazon. So my question, with that, tropical forests can be severely degraded by fire. And then, as I mentioned, with increased drought, forest becomes drier, fire can penetrate the forest, and then there is high tree mortality. That's what we've been measuring. And then, so my question, I want to know the biogeochemical consequences of all that, right? And then, my focus on my PhD was how nitrogen fixation in the soil respond to fire in those situations. And then with more fire, we can have more nitrogen losses into the atmosphere, but then you can also have a release of phosphorus that is so limiting to those ecosystems. And then that maybe be a combination to increase nitrogen fixation activity in the soil. And we're going to see soon. But then first, I'm going to step back a little and explain why nitrogen, right? So nitrogen is, a bit of a paradox because it's abundant, but it's not biologically available. About 78% of the atmosphere is composed of nitrogen gas, but is not biologically available, right? It needs to be fixed, right? So this triple covalent bond needs to be broken to be biologically available, and plants need nitrogen to grow, right? We all need nitrogen. So biological dinitrogen fixation, or BNF, is the largest source of nitrogen inputs to terrestrial ecosystems, right? But it has a high metabolic cost. Okay, so it's costly. It's a costly process. And there are different pathways, right? Symbiotic, and it's kind of the most famous that people, focus more, I would say. Research is more focused on symbiotic. And then we have this free living or asymbiotic that can be, in the soil, in the litter, on the tree barks, on leaves, etc. And we have associatives. For instance, sugarcane is very common to have this, nitrogen fixers hanging out in the rhizosphere. So I focus my research in this free living asymbiotic, right, pathway in the soil. And then it's kind of funny, to say that, but we still don't have enough measurements of baseline biological nitrogen fixation across the world to support, decent representation of system models, right? So this is a recent data synthesis, global data synthesis, and the dots show where the measurements are, right? So I'm highlighting here South America, which is what, 5 points, right? So, you know, it kind of coincides with many things, right? So I met the author here, T. Davis Bernard, and I was asking her in her global synthesis if she tried to use other languages in her search, right? To see if there is research, maybe published in other languages. And then, yeah, she confessed that she's only used the English. So whenever the publication was not in English, she was not able to incorporate. That's why I say like global. But being from Brazil and having done some sort of literature review on what we know about that in Brazil, I say that it is scarce. So we still need, like empiricists, still need to generate data, right? Especially in areas in the world that are not North America or Western US. So my study, I looked into fire effects in asymbiotic nitrogen fixation in the soil in southern Amazon forest, right? At the intersection of where the savanna meets with, you know, the Amazon forest. And for that, I went to a reserve. So it's a protected area, it's a park. And I took advantage of forests that had been well studied for many years. So scientists have been going there since 2004 to study forest dynamics. So these stands are part of the rainforest network, and the vegetation is well studied, but they didn't know anything about soil or the biogeochemistry. So they know that the vegetation is changing, but they can't relate that to how nutrients and carbon, et cetera, are changing too. Right. So that's, you know, kind of my role to add information to the team and also benefit from their knowledge of the forests. So I went to 5 forest stands. You can see here the 5 dots. And this mapping shows the fire frequency between 2000 and 2013. So this mapping had been done previously by a student. And then so the darker areas, the darker red, indicate areas where fire happened several times between 2000 and 2013, up to 8 times. And the light yellow ones is where fire did not occur or occurred just once or twice. Okay, and this is what I used to have this fire frequency gradient in my study. And then within each stand, we noticed another detail that remote sensing can't capture, right? So remote sensing has a 30 by 30 resolution. But when we were in the forest, we saw that fire did not penetrate equally, right, in each stand. So we had this fire presence level of detail that we could only determine when we were in the field. So part of the stand was burned and part of the stand was not at all burned. And we know that because we had the vegetation information. So I created this other level, this categorical level in my study to separate areas that were burned and areas that were not burned. And then, so I sampled soils thinking about not only the fire frequency gradient, but also this fire presence within each stand. And just to show you a little bit what the forest looks like. So this forest, they're seasonally flooded. So when I was thinking about this project, I thought that these fragments, which are natural fragments of forest that occur on drainages, they're going to be the first to go, right? Because they're very susceptible. They're islands of forests. They're surrounded by savanna type vegetation where fire is a constant disturbance, is present. And then they have thin barks, right? So they are very susceptible, especially if they get drier and drier and drier. And this is, you know, just to show the symbols of the sites and the fire frequency gradient. Okay, so, you know, all this sampling was to measure nitrogen fixation in the soil, and I'll explain you next how I did it. And also trying to measure predictors that are cheaper to measure and easier to measure, so we can try and predict nitrogen fixation in the future so people don't have to measure it, right? That's the kind of the main goal here, and that I'm still trying. So many predictors that are known to influence this process, but without exhausting the whole list, right, were the ones that were feasible within my budget. And then doing any type of statistical modeling with nitrogen fixation is like super hard. So this GAM, generalized additive models, they have been working well for this type of data. Okay, So I don't know if anybody here has done any type of nitrogen fixation work, but let me tell you, it's not so easy to measure either. So classically, people have been using this acetylene reduction assay, right? So basically they use acetylene, the reduction of acetylene to ethylene as a proxy, right, for nitrogen fixation. And there is this theoretical 3-to-1 conversion factor. But many studies have shown that this conversion factor varies widely. You know, you can make all sorts of mistakes that you won't know unless you have some sort of calibration. And then so this calibration is possible through the use of tracer experiments, right? Stable isotopes. So that's what I did. I first compared both methods to see which ones I could better rely on for a more robust sampling. So basically this tracer experiment, it means adding labeled dinitrogen gas, right, to vials and compared to vials that did not receive any of that gas, and calculate how much of this labeled gas can 1 gram of soil fix. So it's basically this mixing model, right? So you add the tracer, it mixes with the background, and then you have the sample, and then we have a way to calculate how much it came from the tracer, right? So very different from what I expected. Nitrogen fixation actually was lower with fire, right? So fire frequency did not, the fire frequency gradient I had no significant effect on nitrogen fixation, but the fire presence that I explained to you about had a very important difference. So the top soils, right, the top centimeters where we expect fire to be more important, the burned soils, they had lower, like up to almost 25% lower fixation than the other ones, right? So it kept me thinking, like, why? It should be the opposite. But we're going to go back to this and you'll better understand. So when I tried to put those predictors, right, they're easier to measure, cheaper to measure, we could explain a bit less than 50%, almost 50% of the variability in the data, but we found some very differences between the burned and the unburned samples. So these unburned samples, we found a linear relationship between the total carbon to nitrogen ratio in the soil and the available phosphorus. But these burned samples, it's like we don't see a very clear relationship, right? We see a region where, the yellow where high nitrogen fixation is can occur, and the other area that is, red and low, right? So fire adds more complexity when we're trying to predict nitrogen fixation, right? Compared to areas that we did not have any evidence of fire being present. Okay, so when I was in the field getting this, final sampling for fire, we saw a wildfire happening. And then, I was thinking about what am I missing, right? Because I measured nitrogen fixation in some of those forests years after the last fire event occurred, right? So I wanted to know what happened right after, right? Because maybe there is an effect of, secondary succession. And then also in my previous study, for those who work with fire, you already have a question about fire severity. I'm already mentioning that this observation study, we did not have a way to know exactly how severe each fire event was, because that could be something that, add even more complexity. So in the lab, we could, manage all that. So I brought samples to the lab to burn them experimentally and be able to know what happens right after a fire event. So we put soils in some cups, right? And we measured temperature, and then we controlled for how long we burned, right? So if you think of fire severity being, affected by how long the fire occurs and then how hot it is. So we controlled for time, and then we measured temperature to see the influence of temperature. So we burned them and then measured temperature on the surface, 1 centimeter below surface and 2 centimeters below surface. And then we waited until they cooled down and then did a bunch of measurements, including nitrogen fixation and the other predictors that are easier to measure and might be helpful when we try to predict nitrogen fixation. And I would like to acknowledge the mastermind behind, putting these cups together, Jamie Wright, your soon-to-be doctor. She was extremely helpful. She was at the time a lab assistant and, you know, worked with me for months. So if you talk about nitrogen fixation for her, she's like, no, sorry, not for me. So many hours in the lab to get all this done. So she also put this nice figure together. So this is to show you how temperature changes. So here in the y-axis, we have time in minutes, and the the x-axis time in minutes and y-axis temperature in degrees Celsius. So these lines here you see is at 3 minutes. So we only burned for 3 minutes and then let the samples rest until they achieve their initial temperature. So these different colors here for the fire frequency gradient that I maintained here for this study, but not including all the 5 forests, but just 3, just the you know, both ends of the gradient and one in the middle. So just wanted to note here that, you know, 1 centimeter, just like 1 finger below the surface, is enough to dampen the temperature rise by a lot, right? So the surface, we had temperature going up to 860 degrees, but the 1 centimeter below went up only to 120, right? So, you know, the role of soil, it's just very important. And then, so I'd like to highlight too, that the hotter the temperature, the more phosphorus became available, right? That burned 5 times in my gradient. Did I freeze?
Yeah, from
where? In the past 20 minutes?
No, hotter the temperature, the more phosphorus.
Yeah, that's all you need to know. That's good. Okay, continuing. The last thing I wanted to note here is that this, you know, forest 5 here, which is the one that burned 5 times in my fire frequency gradient, it held a higher temperature for longer, right? So it took longer for those samples to have the temperature drop, right? So there may be this legacy of fire happening, and we don't know if there is some charcoal or something that is hold on to temperature longer. That could be something that mechanistically be influencing nitrogen fixation. Okay, so what I found is that, you know, the immediate response of nitrogen fixation to fire is influenced by fire frequency differently from what I found from my observational study. So these left panels here are from the, you know, the control, you know, non-burned forest. And then when we compared the sample that was, you know, just stood on the bench without being burned by us versus the one where a fire was, you know, where we, you know, put fire in the lab, there was not much change or even a decrease. However, when we looked at the other samples from the higher end of the fire frequency gradient, we always saw an increase in nitrogen fixation right after the fire occurred, right? So there is a difference when you measure nitrogen fixation, if you find higher or lower rates, right? So something that needs to be further explored. So based on what I measured, the hotter the fire, the more phosphorus became available. And then we also found higher nitrogen fixation rates with more phosphorus availability. So we're starting to better understand, you know, what's behind the effects of fire nitrogen fixation starting. But we still, when we try to predict, all those R squares are very low, right? So it's complicated. So summary on this fire portion of the talk is that burned soils actually fix the less nitrogen from the atmosphere in the long term, and the predictable ANF in burned but not in burned soils, right? So fire adds more complexity when we are trying to predict nitrogen fixation. And then fire legacy influenced the immediate fire response of nitrogen fixation. And then, as I mentioned, higher temperatures are positively related to phosphorus availability and nitrogen fixation. And then you would expect them to die, right? These nitrogen fixers, so they're immortal. So, and then the next step here, we also have samples from grassland, the nearby grasslands that I showed you before in that coexistence figure that I haven't added yet to the analysis, and that we want to see how this, you know, nearby grasslands and savanna soils respond to those forest soils, how differently or how similar, as we expect these savanna ecosystems to shift to encroach right into this forest islands. Okay, so now we're going to talk about the climate change and then something that is near you. So you're going to know more about those systems than I do. So the motivation for this study is that we still don't have much empirical evidence for the response of nitrogen fixation in the soil, and not only but nitrogen fixation, but biogeochemistry and plant functional composition to warming drought, right? So we're still, you know, far from fully understanding. And I try to avoid showing this before, but here I'm showing that, you know, there are multiple variables that can influence nitrogen fixation in different directions, right? So if we expect that higher temperature or altered precipitation regimes may influence nitrogen fixation in one direction, well, maybe this, you know, climatic changes may influence other biogeochemical controls on nitrogen fixation that may make it shift into a different direction, right? So it's not so simple to think about one single direction that we expect nitrogen fixation in the soil to change. That's why I'm putting this dashed line here as uncertain, because we are, you know, starting to understand that. So in this project, the goal was to quantify the experimental warming and drought effects on not only nitrogen fixation, but on soil plant atmosphere nitrogen interactions, right? So I took advantage, of course, of this study that started years before I started my postdoc. So here I list below here many of the publications that, you know, I looked into the multiple effects of this experimental warming and drought on plant communities in these prairie systems. And then so I went sort of like towards the end of this project to, you know, do the final sampling before the plots were taken down. So there are basically three sites. One is the northern site is actually central Washington. The central site is, you know, close to you in Eugene. And the southern site is southern Oregon, almost in California. So in each of those sites, there are, you know, different treatments. There are the climate treatments, which are the control, you know, just plots under ambient conditions. There is the warming experiment where there was an increase about 2.5% degrees Celsius. And then the drought where there was a 40% reduction in precipitation. And then besides that, there were plots that tried to represent a high plant diversity, which meant that they were manipulated. So they were seeded with native species. And this low plant diversity actually were just, you know, whatever the community was when these plots were established, right? So, but in fact, this high plant diversity treatment, the diversity is higher. So I took size samples, and then measure nitrogen fixation and many of the previous controls that I had been measuring, except that now I measure these inorganic nitrogen pools of nitrate and ammonia, and also had the benefit of all the plant community data that many others worked for so long, right? And so I'm very thankful for that. It helps a lot to understand not only the soil, but the ecosystem as a whole, if you have information about the plants too. So I'm listing here the ones that I'm thinking about, which is just the plant cover, and then how much legumes there are relative to the whole cover. So we can try to link about potential nitrogen fixation by plants with what the soils are doing. And then also how diverse and then species richness of the plant community. And also many soil and canopy temperature measurements were taken throughout that time that I already have the data provided by Paul Reed and others, and also the volumetric water content in the soil, okay? So I'm sorry to say that the drought and warming effects of nitrogen fixation really depend on where and when the measurements are made. So we were trying to, you know, do this like regional is like, we need to be careful. So first I'm looking at drought here. So on the x-axis, we see log change in nitrogen fixation with the 95% confidence interval. So these lines here represent ambient conditions. Everything to the left means that there was a reduction, and then everything to the right of the line means there was an increase. And then I did these measurements in different seasons. Basically, because in the spring, previous nitrogen measurements show that in the spring there is no nitrogen in the soil because of plant nitrogen uptake. And then in the fall, the nitrogen is back, right? So I wanted to see if that alone could influence, right, the response, and it did. And then, so there is a difference in this high diversity and low diversity plots, right? So for drought, this high diversity plots, we saw many negative effects, right? Like here in the southern site, the effects were negative both in the spring and in the fall. But in the central site, only in the spring was marginally significant. In the fall, not significant. And then in the northern site, in the fall, there was a negative effect, but not in the spring. Like I mentioned, depends on where and when. And then for these low diversity plots, there were less negative effects, right? So for this, the plant communities as they are now, without any type of manipulation for seeding, for increasing the diversity, they seemed more tough, let's say, to drought in some way. So when we look at warming, we don't have the low diversity treatment, so just the high diversity. And then the effects, they were negative also, in the fall in 2 sites, but not in the southern site. And then again, depending on where and when. But based on my data, it seemed like the northern site and the southern site, there was a higher temperature sensitivity of nitrogen fixation. In the central site, all the responses were either non-significant or marginally significant. So, Eugene is sort of like, I don't care about climate change, something like that. Okay, so not to overwhelm you with like a bunch of variables, but I started to explore relationships between nitrogen fixation and everything else that we had available. Like normally we don't find many significant relationships here, but we do find some significant relationships. And then I just wanted to mention too that moisture in the soil was negatively related to everything temperature, right? So when we think about increasing temperature, at least, in this experiment, we see that, soil moisture was like, it was a negative correlation. So the same here. Okay, so did I mention enough that predicting nitrogen fixation in the soil is hard? If not, here I'm mentioning again. So what I did, I took all those variables that I saw that had some sort of relationship with nitrogen fixation and trying to fit different, predictive models. And then I found that combination of soil moisture and the relative legume cover, right? So the cover of legumes relative to the absolute cover in each plot, there was a significant effect on nitrogen fixation. And then I divided that to see how the relationship occurred in each of the treatments. And then, so for the control, it seemed like soil moisture mattered more. So where yellow here is the prediction of higher nitrogen fixation. So higher moisture, higher nitrogen fixation prediction in the control plots. In the drought, it's a bit messy, but in the warming, I found interesting because it was more a connection between soil moisture and relative legume cover, meaning that where both were higher, that's where nitrogen fixation predictors were higher. Okay, so this was just preliminary stats, really, because now I want to do some machine learning to incorporate a bunch of predictors and avoid overfitting, right? Because we have 120, samples, data observations, so it's not too much, right? But we have many, many possible predictors. So I'm going to play with that. And then the challenge is to overcome this complicated mix of processes and variables influencing nitrogen fixation, right? That's something that no one in the world has nailed down yet. And then we do have the frozen DNA extract for all those samples, right? So that is a great opportunity to see if what we see in function matches the composition, right? And the possible changes in not only the composition, but the structure of the soil diazotroph community. So if you have funding, we have those samples. Okay, and then so finally, I'm interested moving forward with research to think about sustaining tropical forests in a changing climate. The situation in the Amazon is very, very concerning. So I'm working on a proposal to try and better understand forest dynamics and help management decisions with that. So taking into account climate change and disturbances, which species are doing better with the drought that we're seeing there. But I also see, that we still need to make more nitrogen fixation measurements because right now there are like these large scale models use functions of net primary production and evapotranspiration for biological nitrogen fixation, and their empirical evidence shows that it's not appropriate. So one of the pressing questions that I know the modelers want to know is what are the energetic and phylogenetic costs and trade-offs of maintaining the ability to continue fixing nitrogen, right? So with that, I want to say many thanks, many thanks to many people, my current employer, the Energy Tropics Project and DOE. Thank the SPA Lab for having me for about a year and something. Bridget Bohannon, Maguire Labs, and then the entire Institute of Ecology and Evolution. I had a great time when I was in Oregon. And then thanks previous labs I was in Davis, and field and lab assistants like Jamie Wright, Hillary Dawson, Toby Maxwell, etc. And just, continue to highlight, you see this fire picture here. So when I was in the field collecting samples for that fire story that you heard, we accidentally burned our car. So in the middle of nowhere at 4 p.m. So, and we didn't have a flashlight. So, doing field work is hard, generating new data is difficult. It takes time, it takes money. And then many times we fall behind in our publishing and everything. So I just want you to keep that in mind. And then whenever you're able to just download the data product on the internet, to just try to acknowledge all those that generated that data, because it is a lot of work, okay? And with that, thank you very much. And I'm sure you have some questions or comments, and I'll take notes and hope to be able to answer you. Thank you. Should I stop my sharing? Or I don't know if people want me to go back to some slide, or I can't hear anybody. So it's like, are you there?
There is
I'm here clapping here, left and right. Thanks again.
You're muted, so I can't hear anything.
Thank you for the great talk. I think we have a question in the chat. Peter, do you want to read that?
Let me see. I don't even know where the chat is.
Go for it, Lucas, unless you want me to.
Yeah.
Okay, I'll do it. Ashley asks, hi, Barbara, is the high end loss during fire from plant biomass volatilized during the fire, or do soil denitrifiers get stimulated in some way by fire?
Yeah, that's a good question. So my first insight of that was because of the first, right, of plant biomass and also soil organic matter, right, being volatilized. Right. So carbon and nitrogen, being burned, they have these gas phases. So whenever we measure that in the lab, when you burn stuff, you lose this organic material. So you lose carbon and nitrogen, and you can lose fire too, right? Phosphorus too, right? When I mentioned that phosphorus availability can increase, but you can also have phosphorus loss because of the particles that fly away, right? With like fire and wind especially. But I don't know about soil denitrifiers, right? This is a good question. I haven't looked much into that. But then I think increasing temperatures, I think you can stimulate a lot microbial activity in general. And then I wouldn't be surprised if the nitrifiers got stimulated. Yeah, but I haven't looked into that. But yeah, good question. That's Ashley. Hi. I can't see you here in the...
Hi, Barbara. I would have asked it, but I was trying to follow directions and ask things in the chat. Thank you for...
Thank you. Yeah, good question. I don't know about the soil denitrifier. It's a good question. Should I read the other question?
Sure, go for it.
Monica is like, not a question, but it's good to hear that fire reduces the nitrogen fixation in the long term, because in Borneo we have a hard time to stopping the burn of the peatland because of the common belief that fire make the soil more fertile. Yeah, this and more reason to less fire. Yeah, yeah. And then even in my measurements, the observational measurements, the availability of phosphorus was lower in these burned soils. Yeah, so especially for forests that are somewhat associated with water, right? So after a fire event, if flooding comes, then it can carry away, right? All that became, it got released after fire. So yeah, the net effect from my observation study was a loss of phosphorus. Yeah. Yeah, it's a pulse of nutrients you can have, as I showed in my experiment. You have this pulse of phosphorus availability, but what happens in the long term, right? It can vary widely, the trajectory. In my case, it got, there was a reduction. Did Jamie send me? Okay, no.
Can I ask a question?
Yes.
Hi, Barbara. Thanks for the great talk. Very fascinating work. I'm curious about the difficulty that you mentioned earlier in your talk of getting a global picture of nitrogen fixation or nitrogen fluxes from the soil, and I guess other compartments as well. And I wonder whether it's at all possible or practical to estimate nitrogen fixation rates through some sort of inverse transport modeling, like people do, for example, for methane fluxes or CO2 fluxes. So you basically measure N2 concentrations on a sufficiently fine grid around the world with sensors. And then if you have a good model for atmospheric transport processes, you could then sort of back calculate to see how much N2 flows into or out of the soil. Is other difficulties of doing that for denitrogen?
That's a great idea that I haven't heard yet. I mean, for nitrogen fixation conversations, at least, people are still very much stuck into, we need to go and measure, right? And then try to find ways to predict, right? So measure other stuff too that is related, and then not having to later on rely so much on having to measure. But I haven't had anybody suggest anything you suggested right now. So you're basically saying trying to track nitrogen N2 flowing into the system as a nitrogen fixation.
Well, you would only get the net flux from something like that. So you would get the net influx or outflux of N2 into the surface from the atmosphere. That would also include fixation at trees, not just soil. So it wouldn't be specific enough to tell you that it's coming from the soil. It could be coming from trees. I mean, one difficulty I can see perhaps is that there's so much N2 in the atmosphere that you'd need very accurate measurements of N2 concentrations in order to get the proper signal to do this. Perhaps with methane, it's different. There's much less methane in the atmosphere. So perhaps it works better for these examples. It's just I've never looked into it, so I'm just wondering whether anybody ever tried it.
Yeah, no, I found it fantastic, but I haven't thought about it. I'm trying to wrap my mind around it. Yes, no, it's definitely something to think about. I can't say if it would work or not because I really don't know.
Okay.
But it sounds like anything that would help us understand without having to measure the entire land surface of the planet would be a good idea.
Well, you'd basically replace the need for doing rate measurements with the need for doing concentration measurements in the air. And if that is substantially cheaper and easier, maybe you can do sort of continuous measurements with the appropriate sensors, and then there would be an incentive to try it. Otherwise, if it's expensive, then maybe it's not worth it.
Interesting. Very interesting.
I think the problem would be that the high concentration of nitrogen in the atmosphere already, and the flux is coming out of the soil, and the canopies are very low compared to, as opposed to the methane case, right, which is very low concentration in the atmosphere compared to the large fluxes coming from the surface.
Yeah.
Because in that case, I don't think it would work. Quick one, Barbara. Can you tell us in one sentence how do people estimate global nitrogen fixation right now? Is it all still legume cover? Is that the main proxy for nitrogen fixation that people use in these models? Is there
So in this large scale models is either net primary production or evapotranspiration. And then this global synthesis that T. Davis Bernard did, she also, of course, compiled the nitrogen fixation measurements, right, from papers, and then also had some sort of NPP or ET value for those places too.
Sure.
And then she found like a zero relationship, like zero, like
So
not even a little bit.
There's no microbial processes in the soil really essentially being added.
So yeah, so that's the thing.
Right?
Yeah, so they're not incorporating exactly the different pathways. It's like biological nitrogen fixation in that forest over there, like the total.
Sure.
Yeah, not breaking up. Yeah, no, not separating which, like
we don't
from where. No, and then so there is a recent paper, like one person in this group that I'm, part of, discussing like ways to improve that. They have a new proposed like equation that is more complicated than just using NPP or ET, right? But that was just brought into the conversation and I didn't look specifically, but it's a longer equation. So it takes into account like temperature and other things that not only just NPP or ET. But then again, what I see from the modeler side, they are careful not to add too much complexity at once, and then I don't exactly understand how to explain why they manipulate these things also, because otherwise it goes all over the place, their simulations. So they're careful into it's not like statistical modeling here, we just add a bunch of stuff and then we play around. So they're yeah, we're still stuck. Basically.
Cool. Well, thanks for the answers. Let's take a quick break here, give you time to drink some water and catch up and come back for more conversations. For those of you who are interested in talking more with Barbara and having other questions related to the talk or otherwise, please